Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
You know the narrative. It's the standard third act twist in almost every outbreak movie, and frankly, it is the scenario that keeps virologists and epidemiologists up at night. We call it the Superbug paradox.
It's the Godzilla of the microscopic world, right? The idea that what doesn't kill you makes you stronger.
Precisely. We invent a new weapon, a miracle drug, a groundbreaking antibiotic. We fire it at the virus, the pathogen, it takes the hit, it stumbles, and then it changes, it mutates.
And what emerges isn't just resistant.
No, it's stronger, faster, meaner. It becomes this unstoppable biological force.
And that is the conventional fear, and for good reason.
With a lot of antibiotics and, older generations of antivirals, that is historically what we've seen. We associate resistance with an upgrade.
We assume that if a virus survives our best shot, it must be because it evolved into some kind of superior predator.
Right.
But, and this is the hook for today's deep dive, what if I told you that that whole narrative is wrong? Or at least in the specific high-stakes case we're about to examine, it is completely backwards. It's a fascinating inversion of the rule. We're looking at a biological trade-off that it just changes the battlefield entirely.
We're looking at a scenario where the virus does mutate to survive the drug. It pulls a Houdini-act to escape the trap.
But the price of that magic trick is just devastating to the virus itself. It survives, sure, but it cripples itself in the process.
It becomes a shadow of its former self.
It's the classic Pyrrhic victory. You win the battle, but you lose the war because you've, destroyed your own infrastructure to do it.
Today, we're unpacking a brand new study just published this month. That's January 2026 in the journal Science Translational Medicine. It comes from a research team led by Nina Penitzdorfer, and it focuses on the molecular wrestling match between the HIV virus and a drug called Lenacapavir.
And this isn't just a medical review. It's a detective story about geometry. It's about structural engineering on a nanometer scale. And it is about the incredibly high, let's call it a tax, that nature charges for survival.
So we're going to explore this paradox of resistance. We're going to meet a specific mutation. Its name is M66I. And we're going to find out why breaking a virus's legs might be the best strategy we've ever had.
It fundamentally changes the game theory of antiviral treatment. It suggests we could force the virus into a corner where its only option is self-sabotage.
So let's set the board. Before we get into the mutants and the broken legs, we need to understand the weapon itself, Lenacapavir. We can call it Len-N for short. What makes this drug a first-in-class inhibitor? I mean, that sounds like marketing speak, but I know there is real science behind that label.
It does sound like marketing, but scientifically, first-in-class is a heavy title. It means it hits a target that no other drug has ever successfully hit before in a clinical setting. For decades, we attacked HIV by going after its enzymes.
The tools it uses.
Right, the tools it uses to copy its DNA, things like reverse transcriptase or integrase.
So it's like kicking the wrench out of a mechanic's hand so he can't fix the engine?
Precisely. But Len-N ignores the mechanic entirely and goes after the car itself. It targets the capsid.
The capsid. Okay, let's visualize this for everyone listening. When I picture a virus, I usually picture that spiky ball we always see on the news. Where is the capsid in relation to that?
So that spiky ball is the envelope, the outer wrapper that it steals from the host cell. The capsid is inside of that. I want you to picture a cone, a beautifully geometric cone-shaped container made of a protein lattice. It almost looks like a piece of modern architecture built from these hexagonal and pentagonal tiles.
Okay, so a protein cone inside the ball. What's its function? What does it do?
It's the armored car. Its job is to protect the most valuable cargo in the world, the viral RNA, the genetic blueprints for the virus. The capsid has to carry these blueprints through the hostile environment of the cell cytoplasm all the way to the nucleus, to the command center.
So it's a delivery vehicle.
A very sophisticated one. It has to be stable enough to protect the cargo, but, and this is key, it also has to be unstable enough to open up at the exact right moment. And here's where LN is so genius. It attacks this vehicle at two completely different points in time. It's what we call a biphasic inhibitor.
Biphasic, 2 phases. Okay, walk us through phase 1.
Phase 1 is entry. When the virus gets into a new cell, that armored car, the capsid, needs to open up. It needs to disassemble to let the blueprints out so they can be copied and integrated into your DNA.
Oh,
Gas binds to the surface of that capsid, and it acts like superglue. It stabilizes the structure too much.
it jams the lock, makes the safe unopenable.
Exactly. The capsid can't open. The blueprints are trapped inside the armored car, and the infection it stalls out right there. The virus gets in, but it can't unpack.
Okay, that's offense. What's phase 2?
Phase 2 is defense, or maybe a better way to put it, is stopping the next generation. So let's say a cell is already infected. It's churning out new viral parts. These parts need to assemble into new capsids to go out and infect other cells. LN binds to the building blocks, these GAG polyproteins, and forces them to assemble incorrectly.
So instead of building a sleek, new armored car, they're building what?
A pile of scrap metal, distorted, open tubes, misshapen cones. These defective capsids can't protect the new genetic material, so the new viruses being released are duds. They're non-infectious.
It prevents the old safe from opening, and it makes sure that all the new safes are built with holes in them.
Exactly. It attacks the life cycle coming and going. And because of this potency, LEN has been a massive breakthrough. It's approved for people with multi-drug resistant HIV. These are patients who have run out of other options. And also for pre-PAP, pre-exposure prophylaxis.
And the dosage, I read this and I had to double check it because it just seemed too good to be true compared to historical treatments.
Twice a year.
That is just wild. I mean, for decades, HIV treatment was synonymous with the handful of pills, the daily ritual.
It's a revolution in what we call pill burden. Just one subcutaneous injection every 6 months. It frees the patient from that daily reminder of their status. It reduces stigma, the logistical hassle.
Yeah.
But you knew there was a complication coming.
There's always a complication with HIV. It's the Houdini of viruses. It always finds a way out.
It is. HIV replicates at a ferocious speed. We're talking billions of particles a day. And it's sloppy. It doesn't have a proofreading mechanism, so it makes mistakes when it copies its genes. We call those mistakes mutations.
And most of those mistakes are duds, right? They kill the virus.
Most of them, yes. But every once in a while, a mistake happens that accidentally changes the shape of the lock just enough that our drug key doesn't fit anymore.
And that is the birth of resistance.
And that is the moment the study focuses on. Penetstorfer and her team wanted to understand exactly how HIV escapes this capsid lockdown. They didn't just model this on a computer, they went to the source. They used samples from 2 major clinical trials, the Capella trial and the Calibrate trial.
I want to pause on those trials because the difference between them seems really important for the context here.
That's crucial. Capella involved heavily treatment-experienced patients.
Yeah.
These are folks who've been living with HIV for a long time, maybe decades. Their virus has seen every drug on the market and has learned to evade them. They have very few options left.
So these are the veterans. Their virus is battle-hardened.
Correct. Calibrate, on the other hand, was for treatment-naive people brand new to therapy. So this gives us a whole spectrum of viral evolution to look at.
Now, in the notes, it mentions that resistance didn't just pop up randomly in everyone. It clustered in a specific scenario they called functional monotherapy. That sounds like a contradiction. Mono means one, but therapy usually implies a whole regimen. What's happening there?
This is the danger zone. Functional monotherapy means that even though the patient might be swallowing, say, 3 or 4 different pills, or taking the shot plus pills, only one of those drugs is actually doing any work.
Why? Are the other pills duds?
No, but the virus might already be resistant to the other drugs in the cocktail. So if a patient's virus is resistant to drug A and drug B, and then you add Lenacapavir, well, effectively, Lenacapavir is the only thing fighting the virus.
Or maybe the patient isn't taking their other daily pills consistently, but they still have the long-acting Len in their system from the shot.
Exactly that. So Len is left alone on the battlefield. The goalie has been pulled, the defenders are on the bench, and Len is facing the entire viral swarm by itself. And when you leave a drug alone with HIV, the virus, it eventually solves the puzzle. It takes time, but with billions of replications, the virus will stumble upon the solution.
So the researchers have these patient samples where Len failed, the virus broke through. Now they want to see why. How did they do that? I mean, do they just look at it under a microscope?
You can't really see these mutations under a standard microscope. They have to do molecular surgery. They isolate the GAG gene from the patient's virus.
The GAG gene, that's the one responsible for the capsid, right? The blueprint for the armored car.
Correct. It stands for group-specific antigen. They take that gene and they clone it, they splice it into a laboratory strain of HIV called PXXLAI.
So they're building a Frankenstein virus, the body of a standard lab virus, but the heart, the capsid, is from the resistant patient virus.
Precisely. This lets them test the resistance in a controlled environment, free from all the other variables in the human body, like the immune response or other drugs. They run these Franken-Viruses through assays. And this is where the detective story takes a twist regarding the tools they had to use.
Right, the distinction between the MT2 assay and the Revlin assay, this felt like a major plot point in the paper, almost a eureka moment for their methodology.
It really was. The standard way to test if a virus is strong or fit is the MT2 assay. It relies on something called the cytopathic effect.
Cytopathic effect. Break that down for us.
So cyto means cell. Pathic refers to disease or suffering. Basically, does the virus kill the cell? You put the virus in a dish with T cells. If the virus is healthy and strong, it infects the cells, it replicates, and it blows them up. The cells die.
Simple enough. Dead cells equal a live, working virus.
But when they took these LNN-resistant mutants and put them in the MT2 assay, nothing happened. The cells didn't die.
So the test said no virus.
It looked like a flat line. But the researchers knew the viral genetic material was there. They had just cloned it in. This created a confusion. Was the virus dead, or was it just incredibly weak?
So weak it couldn't even punch its way out of a cell in a petri dish.
That was the hypothesis.
Yeah.
The superbug that had evolved to beat our best drug was actually too pathetic to kill a standard cell in a dish. So they had to switch to a much more sensitive tool, the Revlin assay.
Which stands for?
Rev-dependent luminescence. It's brilliant. Instead of waiting for the cell to explode, they engineered the cells to emit light to glow the moment the virus successfully gets in and starts producing a specific viral protein called TAT, or Revlin.
So it's a tripwire.
It's a tripwire, exactly. Even if the virus is too feeble to complete its life cycle and kill the cell, if it can just get in and start the engine, the light turns on.
And what happened when they used the light test?
The dish lit up. The virus was there. It was resistant to the drug, but it was so crippled, so unfit, that it couldn't even register on the standard death test. It was alive, but barely.
And this brings us right to the core of the episode, the findings. They found a specific set of mutations that caused this living death for the virus.
They identified a few culprits, Q67H, K70R, and 74D, but the heavyweight champion, the one that really stood out, was M66I.
M66I, let's break that down. What do the M and the I stand for?
They're amino acids. Proteins are just strings of amino acids. So at position number 66 in the capsid protein chain, the virus usually has an amino acid called methionine, or M. In the mutant, it swaps that for an isoleucine, or I.
Methionine to isoleucine. Okay, to me, those are just words from a biology textbook. What's the physical difference? Is one big and one small, or?
They're actually somewhat similar in size, but their geometry, their shape is different. To understand why this matters so much, you have to visualize where LNN sits. The drug binds into this tiny hydrophobic pocket between two capsid protein subunits.
Hydrophobic meaning it hates water.
Right. It's a dry, greasy little pocket. The drug slides in there like a hand into a perfectly custom molded glove. It's a perfect fit. And that tight fit is what locks the capsid together.
Okay, so we have the glove. Now comes the M66I mutation.
The M66I mutation changes the shape of the pocket. The study specifically mentions it introduces a branching at the beta carbon of the side chain.
You're losing me slightly with beta carbons. Give me the analogy.
Okay, imagine the glove again. The finger of the glove is perfectly smooth inside. Now, imagine you sew a hard, rigid marble into the lining of that finger.
Ouch. That would be uncomfortable. You wouldn't be able to get your finger in.
Exactly. Now try to put your hand, which represents the drug, into that glove. You can't. The marble is blocking you. This is what scientists call steric hindrance. The drug is physically blocked from entering the binding site because the virus has put a bump where a hole used to be.
So the drug just bounces off.
It bounces off. And the resistance is massive. We're talking about a fold change of over 1,000. In some of the samples, it was over 2,500. It means you would need more than 2,000 times the amount of drug to get the same effect, which is just impossible to achieve in a human body without toxic side effects.
So the virus wins. It blocked the drug.
It blocked the drug.
Right.
But here's the cost. That pocket wasn't just some empty space waiting for a drug company to fill it. That pocket is a critical structural joint. It's like the rivets holding a bridge together.
So by putting a marble in the joint to stop the drug from getting in,
the virus destabilizes its own bridge. The capsid becomes fragile. It might assemble too loosely, or it might fall apart before it even reaches the nucleus. The study found that viruses with this M66I mutation had a replication capacity of only 13 to 17% compared to the wild-type virus.
13%. That is a catastrophic failure rate. I mean, if my car only worked 13% of the time, I'd be walking.
It is astonishingly low. Usually with other drugs like integrase inhibitors, when a virus becomes resistant, it might take, say, a 10 or 20% hit to its fitness. It's still operating at 80% efficiency. That's still a very dangerous virus.
It's been operating at 15%.
That's a virus on life support.
The study calls this an unusually high fitness cost. It's like the virus had to cut off its own legs to escape the bear trap.
That is a perfect description. And it wasn't just M66I. There was another fascinating mutation they found called N74D.
I remember this one from the notes. This one involved electrostatic repulsion. That sounds like magnets.
It is exactly like magnets. The drug, LN, has a specific negative charge in one region. The N74D mutation swaps a neutral amino acid asparagine for one called aspartic acid, which is negatively charged.
Negative versus negative, they put it apart.
Correct. So the drug is literally repelled from its binding site.
Right.
But N74D comes with a different kind of cost. It seems to disrupt the viruses. Well, it's social life.
It's social life. That's a new way to describe a pathogen.
Well, the virus relies on host proteins, our proteins, to get around inside the cell. One of the most important ones is called CPSF6. You can think of CPSF6 as the Uber driver that takes the virus from the cytoplasm into the nucleus.
The virus can't drive itself.
No, it needs a ride. It hijacks our cellular transport system. The N74D mutation messes up the interaction with CPSF6. It essentially cancels the Uber.
So now the virus is resistant to the drug, but it's stranded at the airport with no ride to the city center?
Exactly. It can't get to the nucleus to integrate its DNA.
Yep.
So again, you have high resistance, but a very low ability to actually cause an infection.
Then there was the double mutant, Q67H and K70R. These sounded like a dynamic duo.
This is where the virus gets clever with teamwork. Individually, Q67H and K70R are pretty weak. They don't stop the drug much, and they don't hurt the virus much either.
Yeah.
But when they appear together, they're synergistic.
The whole is greater than the sum of its parts.
The study details this intricate network of hydrogen bonds and water molecules that usually bridge the drug to the virus. These two mutations, they work together to disrupt that water network. It's like cutting the suspension cables on a bridge. But even here, there's a cost. It isn't as severe as M66I, but the virus is still compromising its own structural integrity just to survive.
So we've established that the virus is essentially mutilating itself to survive, Elamine. It's weak, it's slow, it's fragile. This leads to what I think is the most important question for anyone listening to this. If the virus is so pathetic, why did the patients in the trial experience viral rebound?
This is the clinical reality check. It's very easy to look at 13% fitness in a lab dish and think, game over, we won. But in the human body, the math works differently.
13% of a billion is still a very big number.
Exactly. If a patient has a high viral load to start with, and they're on functional monotherapy, so nothing else is there to kill the virus, that wimpy virus has an open field. It replicates slowly, yes, it sunstalls, but it does replicate. And over weeks and months, that viral load will climb back up. It might not reach the same explosive peaks as a wild-type virus, but it will be enough to make the patient sick and infectious.
And the virus doesn't just sit there and accept its fate, right? I mean, evolution is a process. The study mentioned compensatory mutations.
Right. The virus realizes, hey, I have no legs, so it starts to try and grow crutches. The researchers found that once M66I was established, the virus would start acquiring secondary mutations, like A105T or T107A.
And do these fix the problem? Do they get their legs back?
They patch it. They don't fix the original problem in the pocket. The drug is still blocked. But they might stabilize the overall capsid structure just enough to raise the fitness from, say, 17% up to maybe 34 or 40%.
So it upgrades from a crawl to a limp?
Precisely. In the scope of this study, at least, the virus never returned to 100% fitness. The damage done by M66I is just too fundamental. It's a permanent scar. But a limping virus is still a virus and allows the infection to persist.
This brings us to the strategy then. The authors of the paper seem to view this fitness cost as a major strategic advantage for us. How do we exploit a limping virus?
This reinforces the absolute critical necessity of combination therapy, the cocktail.
We've known about cocktails since the 90s. Why does this study make that argument even stronger?
Well, think about it. You have a virus that is resistant to Lenin, but it's replicating at, let's say, 1/10 the speed of a normal virus. It's fragile. It's slow.
It's a sitting duck.
It is the perfect target for the other drugs in the regimen. If you're also taking a reverse transcriptase inhibitor or an integrase inhibitor alongside Len, those drugs are waiting. The wimpy Len-resistant virus simply cannot outrun them.
So the resistance acts as a bottleneck. It forces the virus into a shape and speed that makes it really easy for the other drugs to kill.
Exactly. The only time this fails is if the other drugs aren't there. If the patient stops taking their daily pills, or if there's some hidden pre-existing resistance to those pills, then and only then does the wimpy virus get a chance to survive and replicate.
So if Len has teammates, the resistance is manageable. If Len is left alone, resistance is a disaster.
You cannot let the goalie play the game alone. The study authors explicitly state, and I'm paraphrasing here, that these data support the development of treatment regimens that combine Len with other active antiretrovirals.
It's a squad game.
It is. And this also gives us a map for the future. The researchers didn't just find the mutations, they mapped them with atomic precision. We know exactly what that bump in the M66I side pocket looks like now.
So can we build a key that fits the new broken lock?
That's the next frontier. Scientists are already looking for next-generation capsid inhibitors. Maybe a molecule that's more flexible, one that's sort of wobbly enough to mold itself around that bump, or a molecule that binds to a different part of the capsid entirely.
The arms race continues. We jam the lock, they break the door, we reinforce the door.
And on it goes. But what's unique here, and this is what makes me optimistic, is that we force the virus into a corner where its only escape route involves a kind of self-destruction.
It really is flipping the script on the whole superbug fear. It's not that the virus becomes stronger, it becomes desperate.
Desperation is dangerous, for sure, but in biology, a desperate organism is a vulnerable one.
So let's synthesize this for the listener. We've covered a lot of ground here, from protein cones to glowing cells to the mathematics of survival. What are the big takeaways?
Well, for the science geeks listening, it's a validation of the methodology. You can't rely on cell death alone to measure viral fitness anymore. You need sensitive tools like the Revelin assay, because the new generation of resistant viruses might be silent in traditional tests, but still be present.
And for people living with HIV, what's the message for them?
The message is loud and clear. Protect the Len-N. Lenacapavir is a Ferrari of a drug, but you can't drive a Ferrari off-road. You need to keep the road paved by taking your background regimen.
Adherence.
Adherence. If you miss your other pills, you're leaving LN alone in the ring. And that is when M66I emerges. And even though it's a weak mutant, it still opens the door to treatment failure.
It really highlights that resistance is such a dynamic process. It's not a light switch that flips on or off. It's a negotiation between the drug and the virus, and we want to keep the virus negotiating from a position of profound weakness.
Exactly. We want to keep the cost of business too high for the virus to pay.
Before we wrap up, I want to throw a provocative thought at you. You mentioned we are forcing the virus into a corner. We see that M66I makes the virus wick. Are we reaching a point where we can engineer a true evolutionary trap?
Go on.
Well, imagine we design drug A so that resistance to it requires a mutation like M66I something that fundamentally breaks the capsid. Then we design drug B that specifically targets that broken capsid, a drug that only kills the wimpy resistant virus.
You're describing a concept that's it's called collateral sensitivity, or sometimes orthogonal resistance.
Like, does it actually exist?
We see hints of it in biology. For example, some mutations that make HIV resistant to one drug can actually make it hypersensitive to another, older drug. If we can map these trade-offs perfectly, we could theoretically create a regimen where the virus has no good move left on the chessboard.
Checkmate.
Where the only path to resistance is, in effect, total viral suicide. We aren't quite there yet, but studies like Pennistorfer's are drawing the blueprints for that very trap. They're showing us exactly where the structural weaknesses lie.
A trap where the only way out is down. I like the sound of that. It feels like for the first time in a long time, we are getting ahead of the virus's ability to innovate.
We are certainly making it work harder for its survival than it ever has before.
Well, that's all the time we have for this deep dive into the microscopic battleground of the HIV capsid. It's a reminder that even in the invisible world, the laws of physics and, well, economics still apply. You can't get something for nothing. Even for a virus, survival has a price.
A very, very steep price.
If you're listening and you happen to be on one of these new injectable regimens, remember, you are part of this strategy. Taking your full cocktail is what keeps the virus cornered. Don't give it a fighting chance.
And keep an eye on this space. Capsid inhibitors are just getting started.
Thanks for listening, everyone. Stay curious, stay informed, and remember, sometimes the scariest monsters are actually just limping along on broken legs. We'll catch you on the next deep dive.
Thanks for tuning in to the Health Headliner podcast by To Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.